Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from Department of Biochemistry. Today is my 5th lecture, and in this lecture we will study about what is PCR, what is its method, what are its advantages and application. PCR is in vitro method for the amplification of the DNA. It stands for polymerase chain reaction. It is a test tube method for amplifying a selected DNA sequence. PCR permits the synthesis of millions of copies of specific nucleotide sequence in few hours. So PCR takes a very less time as compared to other techniques for the amplification of the DNA, like cloning. It can amplify the sequence even when the target sequence makes up less than 1 part in a million of the total initial sample. It means that it is very sensitive method. It can even detect a very small quantity of the DNA and can do its work, which is amplification. It can be used to amplify sequence from any of the DNA sources, like bacteria, viral, plant, animals. The procedure, the principle of its work is similar for all the DNA sources. DNA polymerase is used to repetitively amplify target portions of the genomic or cDNA. Each cycle of amplification doubles the amount of DNA in the sample, leading to an exponential increase. In the one cycle, from the one DNA molecule, 2 DNA molecules are produced. So in the first cycle, 2 DNA molecules, and in the second 2 to 4, and in the third then 4 to 8, there is the exponential increase. The amplified DNA product can be separated by gel electrophoresis, detected by the Southern blot, and hybridization and sequence. Once we have the DNA product in the sufficient amount, the purpose of the PCR is just to get the DNA in sufficient amount so that it can be used in the Southern blotting, hybridization, and it can be used in the sequencing. So the PCR method is just to get enough DNA. What are the steps of the PCR? First of all, the first step, it is the primer construction. Then is the denaturation of the DNA, annealing, and chain extension. We will see all the steps one by one. The first is the primer construction. Primers are the see that these are the these are the primers, the blue ones. They are the primers. They are complementary to the sequence sequence of the DNA molecule at the 5' and at the 3' end. These are the short nucleotide sequences. So primer, it is necessary to know the nucleotide sequence. It is necessary to know the nucleotide sequence of short segment on each side of the target DNA. So for the primer construction, we should know the sequence on each side. Not all of the DNA sequence is not necessary. The sequence of nucleotide is not necessary to be known, but only a small size. Only a short segment on each side of the target DNA, its sequence is necessary so that we can synthesize the 2 primers. One primer is attached at the 3' end, and the other is attached on the other strand of the DNA at the 5' strand. The nucleotide sequence of flanking regions. Flanking regions means the regions at the both ends. They are used to construct 2 single-stranded oligonucleotide, usually 20 to 35 nucleotide long, which are complementary to the respective flanking sequences. So this is the definition of the primer used in the PCR. Means single-stranded oligonucleotide 20 to 35 nucleotide long, and they are complementary to the respective flanking sequences. These are the primers from which the polymerase enzyme will start its synthesis. Okay, now this is the steps of the PCR. The first is the then after the synthesis of primer, the next step is the denaturation of the DNA. This is the denaturation of the DNA. The two strands are separated by increasing the temperature about 95 degrees C. At the 95 degrees C, the two strands they get separated. Two strands of the DNA they get separated. The next step is the annealing of the primers. In the next step, the temperature is lowered almost about 72 degrees C, and at this temperature the primers of the at about 50 degrees C, the temperature is lowered down to about 50 degrees C. The two primers at this temperature, they will anneal, they will attach to the complementary sequence of the DNA. This is the annealing of the primers to the flanking region of a single stranded DNA. So these the green colored strand, this is the primer attached at the 3' end of the DNA, and this is the primer attached at the 5' end of the at another 3' end of the DNA. So these the temperature is lowered down so that the primer can be attached at the flanking regions. Two primers are attached, one attached at the lower strand and another attached at the upper strand. The third step, this is the extending the primers, extension of the primers. In this, the DNA polymerase, they will uses DNTPs, deoxyribonucleotides, which are added in the experiment, which are added in the test tube, and the temperature is increased to 72 degrees C, which is the optimal temperature for the working of the enzyme. Enzyme used is the Taq DNA polymerase. The Taq DNA polymerases will uses the enzyme and extend the primer, and again the synthesis is from the 5' to 3' end. Okay, the both strands, they are extended in this way and in the direction of the chain growth in this way. So in the fourth step, now we have from a single DNA molecule, we have the two DNA molecules. The two new double-stranded DNA molecules can be then denatured and copied by again going to the step from 1 to 3. So this is the all the steps which are used in the PCRs. This is the first cycle. In the second cycle, this is the first cycle in which the one DNA molecule is doubled, right? From the one DNA molecule, two DNA strands are produced. In the second cycle, from the two DNA molecules, here the green colors, they are the primers, and in the second step, from the 4 DNA strands, they are produced. In the third cycle, from the 8 DNA molecules, they are produced. So this is the exponential increase in the number of molecules of the DNA. Each strand bind a complete complementary primer, and the step of the primary extension is repeated again and again so as to increase the amount of the DNA. And I have told you before that the enzyme thermus aquaticus TAC polymerase is the TAC polymerase is the enzyme which is obtained from the bacterium Thermus aquaticus. It lives in high temperature. This enzyme is particularly used in the PCR because we have to increase the temperature up to 95 degrees C. This enzyme, it do not denature at the high temperatures. So about in 20 to 30 cycles they are run in the process, and 1 million to billion folds of increase in the amount of the DNA it can be obtained, right? So this is the how the DNA it is multiplied, it is increased in the PCR. What are the advantages of the PCR? There are two main advantages of the PCR. The one is the sensitivity and speed. Sensitivity means the DNA of an individual cell can be amplified. It means a very less amount in a single cell. The DNA present in a single cell, it can be amplified. It is so sensitive that even a minute amount of the DNA, it can be detected by the PCR machine and by the enzymes and the all other things present in the PCR reaction, and the reaction can be reaction can take place. And the speed is that the PCR is fast and technically less difficult than the traditional cloning methods. So its speed is fast and it is less difficult. Even in 2 to 3 hours we can have the million and billion fold increase in the amount of the DNA. So advantage of PCR, there are two advantages: sensitivity and speed. What are the applications of the PCR? The first of all, synthesis of mutant DNA in sufficient quantity. When we have to study the DNA, we get the cells from some source. For example, we are getting some samples from a fetus, right? And we don't get a sufficient amount of the DNA from the fetus, which will be harmful to the health. So we get the mutant DNA from the fetus and we'll do the PCR so that we have the sufficient quantity in order to study, in order to analyze that DNA sample for the future analysis, for the further studies. The next step, the next application of the PCR, this is the detection of low abundance nucleotide sequence. For example, HIV virus. These virus, they have a long latency period, right? HIV virus, they have the long latency period and they are difficult to detect at early stage of infection. But the PCR, as I have told you before, that it is a rapid and sensitive method. It can detect the viral DNA sequence even when only a small portion of cells they are present in the human blood. Within the days of the infection, we can detect HIV virus. We can detect any type of the virus in the human blood. The next application is the forensic analysis. PCR is used in the forensic analysis. We have discussed the forensic application of biotechnology in forensic analysis in our previous lecture also. In the forensic analysis, if we get evidence, a single human hair, a tiny blood spot or a sample of semen from the crime scene, it can be amplified, right? We cannot अगर हमारे पास एक कहीं evidence मिला है, एक single hair follicle from the crime scene या एक tiny blood spot मिल गया है, तो अब एक blood से हम उसको आगे कैसे लेके जाना है? अब इस एक ब्लड से हमारे पास जो है कोई एविडेंस नहीं मिलेगा. हम क्या करेंगे, उस ब्लड से या ह्यूमन हेयर फॉलिकल से या सीमन से हम DNA को एम्पलीफाई करेंगे, उसका PCR करेंगे, ठीक है, ताकि हमारे पास एक सफिशेंट अमाउंट में DNA आ जाए और हम फिर उस DNA की प्रोबिंग या सॉदरन एनालिसिस करके या उसका RFLP करेंगे, सो दैट वी कैन हैव द much amount of the DNA for the detection of the criminal. अच्छा, अब नेक्स्ट इसी फोरेंसिक एनालिसिस में पैटर्निटी टेस्टिंग यूज़ द सेम टेक्निक्स. सम टाइम जो हॉस्पिटल्स में जो है वो बच्चे मिक्सअप हो जाते हैं, क्योंकि बहुत ज़्यादा केसेस आते हैं डिलीवरी के, तो उसमें है कि हम बाय यूजिंग अ स्मॉल सैम्पल from the children, paternity testing can be helpful in finding out the or in the solution, in finding out the solution. Prenatal diagnosis and carrier detection of the cystic fibrosis, this can also be helpful by using the PCR. Okay, this is the how the PCR is helpful in the prenatal and carrier detection of the cystic fibrosis. See, this is the normal CFTR gene. In the cystic fibrosis, the CFTR protein is mutated. This is the cystic fibrosis transregulator proteins which is mutated, and the cystic fibrosis, this is an autosomal recessive genetic disease. And CFTR, this is the cystic fibrosis transmembrane conductance regulator protein, and the mutation present in the CFTR, this is the 3-base deletion. It that results in the loss of phenylalanine residues from CFTR protein. So this is the normal CFTR, and this is the mutant, which is a little bit smaller than the normal. So what we do, we will do the PCR of the genes of all the from the homozygote normal, from the heterozygote mutant, and from the homozygote mutant which is affected. So we will get the 3 type of patterns. If in the homozygote normal, the band will be as we know that this is the 63 base pair, इसका band जो है वो upper side पे आएगा, ठीक है. heterozygote में we have the 2 type of bands, ठीक है. एक allele में उसका जो है वो 63 base pairs present है और एक में 60 base pairs present है, ठीक है जी. अब mutant में जो homozygote mutant है, mutant CFTR gene is missing, as you know, 3 base pairs, so it gives a shorter than the band is shorter than the normal product. So this was all about my 5th lecture. Best wishes. Allah Hafiz.